Hello everyone, welcome to Biology in Minutes. Today we'll be talking about protein synthesis part 1. So first off, gene expression. Gene expression is the process where DNA makes protein, or protein synthesis. DNA is the instructions to make proteins. So that's gene expression, but how can DNA actually make protein? Now there are two steps to this process: transcription along with translation. In this video, we're going to be talking about transcription, and part 2 we'll talk about translation. So transcription is the synthesis of RNA using DNA. The type of RNA synthesized is known as messenger RNA. So the process of transcription is, again, we have DNA, and then we turn that to RNA. And notice the difference between these two nucleotides. DNA is double-stranded, while RNA is single-stranded. So now the steps of transcription. First, the enzyme involved with this process is called RNA polymerase. RNA polymerase unzips the two strands of DNA. Next, RNA polymerase joins together the RNA nucleotides complementary to the DNA strand. What these two steps mean is that after RNA polymerase unzips the two strands of DNA, it then adds RNA nucleotides based off the DNA. An important thing to note is that RNA polymerase moves from the 5' to 3' direction, which means it adds to the 3'. Here is the example of the DNA strand. So the first step RNA polymerase does is initiation. In this process, RNA polymerase binds to the promoter. In this diagram here, we see that is the promoter, and RNA polymerase binds to that. The next step is elongation. In this process, RNA polymerase moves downstream. Finally, the last step is termination. In this step, RNA polymerase reaches the terminator, and the messenger RNA releases. It's important to note that eukaryotes have more complicated systems. Eukaryotes don't actually have the terminator that's in prokaryotes, but it's a lot more complex, and that's an idea for a future video. Transcription has completed. However, before we can move on to translation, there is a small step, and it's the modification of pre-RNA. This molecule here is messenger RNA, and it's made from transcription. The mRNA made from transcription is called the primary transcript, or the pre-RNA. The modifications happen when the 5' end of the pre-RNA receives a guanine cap, and the 3' end of the pre-RNA receives a poly-A tail. After these steps are done, the pre-RNA then becomes the mature RNA. The mature messenger RNA is the RNA that moves on to translation. Thank you for watching today's video.